Hi there.
Welcome to Microbial Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aim at the public with the tools of biotech.
My name's Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist. I'm currently working as for a scientific journal.
Every week we meet to talk about microbiology, and today it's a news week where we do an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
And at some point we'll be doing deep dives. So if there's any other papers that we talk about today that catch your fancy, please message us at microtwjc and we'll look into something that you suggest to us.
You can follow along with any of the papers that we discuss in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you. So please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. First, we delve into Omicron and some of the questions as to how it rose. And then we look at it more in depth at how the SARS-CoV-2 interferes with cells through messing with our innate immune responses, and also for how it scavenges lipids from our cells. Then we also get to see about how the link between immune responses in our gut and skin microbiota and how that links everything together. We also get to see how antibiotics based on RNAs can be used to target genes, and how we can create quote unquote bacterial microbots, which can be steered using magnets. All that and more. So you will want to stay tuned.
Yes, excellent. So yeah, as typical, we're opening up with some of the SARS-CoV-2 papers that we've seen in the literature. This first one is called Tracking SARS-CoV-2 Omicron Diverse Spike Gene Mutations, Identifies Multiple Intervariant Recombination Events. And yeah, this had hit the news like a while ago, I think. But this paper, I guess, is recently published, and I thought in the introduction it did a good job too of talking about like some of the, I guess, controversy in the field. So this surrounds the identity of the existence of deltacron as supposedly a recombinant variant of the virus, parts of the Delta variant with an Omicron variant. And in the introduction, actually did a good job of saying that when that was first reported, they thought it could have been from sequencing error. But then this particular study, I guess, has waited a little bit more time to like accumulate more genomes inside of that GSAID database, and then it reanalyzes it to try to, I guess, nothing new in terms of like it waited more time for genomes to be populated into the database, and then trying to find that signature of recombination.
Yeah, because I do remember when this came out, we were like among the first people like were going like, oh well, this could be a bit of an accident. We don't know
Yep.
because we need to wait for those genomes to come out clean, and then we can make something of it.
Yeah, because they have to like it's only the strength of the data that they have in order to make these comparisons. It's all an in silico analysis. So when the reports first came out, there wouldn't have been as many, but now we've experienced a lot of the Omicron wave, and certainly like even several types of Omicron have come. So doing these comparisons between genomes, I guess the associations get a bit stronger.
Yeah, I think we just want to remind people that Omicron almost came out of nowhere. There was like it came out with so many mutations and so different from other things. There was this kind of big mystery about how it could have come about. I think this paper aims to like pull the rug over under, like unveil, see, look at what might be going on.
Yeah, and maybe it's also kind of about the relationship between BA1 and BA2, and looking at like how those two are related, and what sort of mutations they might have in common with other variants, and then trying to figure out whether or not that could have been driven by a recombination event. So I think maybe a quick explanation, right? We can think of variants as occurring through some sort of like natural variation, like sort of replication errors, right? Accumulate some natural diversity, and then those get selected for in the population, and that's how we get certain variants. But the mechanism that they're trying to describe here is that maybe there might be cases where you get co-infection from two different variants, and then and it's being demonstrated in other coronaviruses that those genomes can sort of get jumbled up together. So I mean, that's almost like sex in some ways, right? Like the same way that we have like allelic recombination to generate diversity. If you have enough infections with SARS-CoV-2 out there, it's possible that in that co-infection event, it could happen. And I think we did cover once, like there are some like clinical case studies talking about like immunocompromised individuals who, when they sequence the pool of viruses inside of them, they have like a huge diversity because they aren't able to suppress the infection well. And so there's one hypothesis, again, it's not, we don't really know, but there's a hypothesis saying that maybe that's a site, an infectious sort of event that could generate some of this diversity, and then it gets spread in the population and selected for there.
Yeah, and I think another thing that I wanted to bring up was, oh god, I think oh no, I forgot what I was gonna say. That's disastrous.
Okay. Well, you can interrupt me at any time. I'll try to I'll go into something now, and then just let me know if you remember. So they're looking for a signal of recombination, and so the way that they do that is they only have sequence. And so they chop up the genome into many little pieces. Figure 3 is actually where we might be able to see something.
I remember what I was going to say, because this is actually very...
Oh, okay. Nice.
What you brought up is very difficult, because SARS-CoV-2 genomes are very similar. So if you have two identical viruses and they recombine, you can't spot it very easily. So, so this is a challenge they have, trying to find out where those recombinations are happening. Because they're so similar.
Yeah, and I think that's probably what drove the uncertainty of the first one. There weren't enough like sort of sequenced Omicron infections to get a sense of using this analysis method, they weren't able to maybe confidently identify the recombination. But now there's a lot of Omicron genomes. I mean, that's all we have, or that's the majority of infections these days globally, although there are infections from other variants as well. And so, so they have all these sequences, they chop up those sequences into like overlapping fragments, and then they make phylogenies of all those fragments. And the idea is that like there might be, when you compare all of those different phylogenies, all those different trees, there could be some fragments of genomes that are assorted with one lineage in one tree, but in another tree, like on another, for the same genome, other segments in a different region could be associated with other lineages. So that's the signature that they're looking for in the data that would sort of indicate some sort of recombination. So in some parts of the genome, it has a close association phylogenetically with one branch of SARS-CoV-2, and then in another part of the genome, there's fragments that associate stronger with another. And the idea is that, that it's more, I guess it's more, it's a easier explanation to say that all those mutations sort of came together from one recombination event rather than like emerged all patchworky and then got selected for later in the population. And so they point out that there's they can see like that there's some Omicron variants that have signatures coming from Delta, or even coming from the ancestral strain, the Wuhan, the Wuhan one. And so, I mean, I guess those things are still in circulation. And I guess also it's nice to see because Omicron is so different from the rest, that's also something that drives this analysis because, I guess chunks of mutations could be identified from other viral genotypes.
And I think there's still a lot of mystery there, because I think there were a lot of mutations that weren't identified from the study that we still don't necessarily know the origins of. So I think there's about 30 in BA1 or BA2, I think they mentioned, that they still need to find explanation for. And there's been all sorts of kind of thoughts on maybe it coming from zoological sources of rats, mice, those sorts of things. I mean, I know in the past we've had some kind of evidence about that, where I know there's some people who looked at some genome sequencing and said, hey, this looks like mutations that often would happen in mice or rats, so maybe that could be that's a bit controversial at the moment, but I thought
the authors also point out like the mink, right? and we'd covered that too in the news, that there were instances of seeing mutations that appeared in mink inside of then human isolates. So I guess one of the takeaways from this paper is just, well, first of all, like giving a little bit more confidence in thinking about the existence of recombinants, like it could be happening out there in nature, and identifying that as a source of variation that could help explain, like, if we do see the rise of a new like lineage, and we're seeing like mutations from different sources, maybe it is coming from some recombination event inside of the human population, or inside of animal reservoirs.
Yeah. All right. So we can move on to the next paper. Okay. SARS-CoV-2 N protein antagonizes stress granule assembly and IFN production G3BPs to facilitate viral replication. So,
Yep. Kind of a little detail mechanism of the ways in which SARS-CoV-2 uses its many proteins to get stuff done. In this case, we're talking about the N protein. So the nucleocapsid, the stuff that's coding the genome. They actually find out, they find out that it might be interfering with stress granule formation. So that's like a generalized stress response that happens inside of cells, where upon some level of stress, mRNAs get sucked up into like inclusion bodies, essentially, sort of stalling some translation. And that's thought to be a useful stress response because like you could basically say pause on regular cell like life and then prioritize some other type of translation to help with whatever, to ameliorate the stress in some way. And so in this paper, they hypothesized that NP, I think they had some observation that said the nucleoprotein could be interfering with this in some ways. And so they go into detail to try to figure out whether or not that's happening. So they do like IP to see like, oh, in these stress granules, we see SARS-NP protein is in there. So that's like their first clue into trying to investigate this.
So because they came from like predictions, so they predicted that it was some affinity with G3BP1 and so those are very important for the formation of those stress granules.
Right.
Right. And so they do a couple of, and from there they kind of work it through. This is very like kind of good like logic-based microbiology where you kind of find one lead and then you trace it down and see what else might come out.
Yeah, absolutely. They have a very like targeted hypothesis about like the association between a stress granule protein and this NP protein. And it's like in the second figure, they sort of validate what they think is happening by looking at the infected lungs of mice and people. And they're able to see like in these histology samples some co-staining of the NP protein and the G3BP1.
Yes, and if I get this correctly, the stress granule response is part of like our innate immune response, or even a reaction to stress. So I think you mentioned earlier that it slows everything in the cell down, but also that scuppers at the virus's chances as well if we put the brakes on the metabolism.
Right, and so I think that's part of why the hypothesis is so attractive that SARS-CoV-2 should be doing something to it, because SARS-CoV-2 needs to evade it in some ways in order to be successful inside the intracellular environment. And like this is the classic refrain of so many viruses. They have these multi-functional proteins that do more than one thing. So like NP can cover the genome and help it find its way to packaging or something like that, prevent it from degradation. But it could also prevent these. It is an RNA genome, so it can prevent the genomes from getting sucked up into stress granules because it just like disrupts that natural formation.
Yeah, and I think they have some. They do show. Well, they've got a lot of immunoprecipitation, where they link this these kind of proteins together to see like the degree of binding in different situations. So they even like chop it up into little pieces to figure out exactly where it's binding.
They find like the exact region that gives the association between this stress granule protein and the NP protein.
Yeah, and we've got, so let me see, I think it was, because we got some brilliant figures of this like kind of happening, right?
Yeah. Yeah, the association of, yeah, just of that small truncated bit. And then I guess they try to give it some immune relevance by at the end linking it to um, like preventing some of the innate immune response signaling pathway.
So yeah, a really interesting insight into how the virus yet again tries to manipulates the host to do what it
Yes.
wants rather than what. And I think that follows on from that.
Yeah,
We've got
that's all.
another paper
Mm-hmm.
called Global Lipid Map reveals host dependency factors conserved across SARS-CoV-2 variants. So this paper came across because last week, I think, well, not last week, but the last time we were on the show, we talked about how the SARS-CoV-2 creates these replication organelles in the cell that kind of suck
Mm-hmm.
up double membrane vesicles, and how it kind of takes away some of the lipids, and it kind of eats some of the cell's own lipids and use it for its own kind of creation of organelles. This is another look at
Mm-hmm.
how they basically look at um, host lipids and seeing how that metabolism changes during SARS-CoV-2 infection. Um,
Yeah.
and so there's a lot of
There's a lot of
a
lipids.
lot of different lipids,
I didn't. Mm-hmm.
a hell of a lot of chemistry here that's going on because there's lipidomics. So there's lots. So you've got the one that they're focusing on is triacylglycerol, which is a polyunsaturated free fatty acid, which has been linked to severe disease outcomes in patients. Um, so they kind of they took a look at using kind of chromatography to separate out all these different chemicals to identify which ones are the most changing when cells. And so a lot of
Yeah.
these was done with just like cells in cell culture. Uh,
Yeah, they have two cell types. They infect with SARS-CoV-2, and they compare the lipids that are made in the healthy cells versus the lipids that are made in the infected cells. And they find like some signature of infected cells, right? That
right.
is common between the two cell types. So that lends the authors this thought process that SARS-CoV-2 is doing something to try to enrich a certain set of lipids because it needs those lipids for its own purposes.
Yeah. And once they do that, they try and follow it out. So we know something is causing these changes. What is it? So they
Mm-hmm.
take out each individual like kind of protein and open reading frame from SARS-CoV-2 and
Uh-huh.
test those metabolically to see whether those are causing issues. And
Mm-hmm.
I think they narrowed it down. So the triacylglycerols being caused by ORF6, NSP13, NSP5, and also with ceramide are very important. Like so, we NSP6 was which we talked about last paper also was key for creating the replication organelle. So there,
Mm.
as we're picking up from the last one, SARS-CoV-2 proteins, a lot of them do multiple things. They're Swiss Army knives of they do have a
Yep.
lot of different jobs because it's tough in this economy. Uh, so they
Working hard.
yeah, it's hard for because especially SARS-CoV-2 has a limited amount of protein, so they have to do a lot with that. And they trace this down and they realize, okay, well, we identified these host factors are affected by SARS-CoV-2. What if we messed with those? What if we added inhibitors for those host factors? So they use a couple of inhibitors for like cholesterol production or things that in that like increases or like the values of something. So
Yep.
I noticed that they used orlistat, which is kind of a drug that was created to prevent obesity by by manipulating these very parts of the metabolism. Um,
Yep. Yeah. Maybe get us to figure 5 is like a good visual representation of
yes.
what they're doing. This is kind of like the bringing it home, maybe trying to give some applications, I guess, like potential ways of inhibiting. I think I've seen this similar approach. It came out also during genetic screens. They may have mentioned it in the introduction that like if you do like a CRISPR screen of like host factors that inhibit viral replication, you also get some of these lipid manipulating enzymes, lipid metabolism enzymes. So yeah, it is important, probably because of its participation within these double membrane vesicles, that a specific type of lipid becomes enriched in the infected cells.
Yeah, I mean, it's the interesting part here is that we're finding out these lipids are important for infection because SARS-CoV-2 is going to go to all this effort to mess with them, to create them, or
Yeah.
and so it creates thought of because I'm thinking like linking it to because obesity is often thought of as like a cofactor in causing like severe SARS-CoV-2 disease,
Yeah.
and people generally try to link that to like general health issues. But what if it
Right.
was something specific that was happening with the metabolism that SARS-CoV-2 was taking advantage of in those situations? Would
Sure.
it so? I mean, I think there's a lot of like stuff that gets ignored when it comes to obesity because people get like written off. But I think
Mhm.
there's a lot of interesting metabolic questions that this paper raises.
Yeah, yeah. I mean, I guess that's like, yeah, I'm not being familiar with the obesity literature. Like maybe it's known like there's specific lipid profiles, right, that get changed within obesity, and like yeah, as you say, it could give us like a mechanistic understanding beyond just like a general everyone's health is worse when you're obese, but something specific that it's like interacting with, yeah, SARS-CoV-2 in a certain way.
Yeah. I mean, lipids are important signals for the immune system. So we've got things like
That too.
steroids or other that are very important for the immune system.
Hmm.
And understanding how diseases like SARS-CoV-2 interact with those might yield some. I mean, probably there are loads of researchers working on this right now. So I'm
Yeah.
assuming that this is what they're looking at.
Yep.
It's
And then at
um
the, I'll say also like, even if it doesn't produce any like therapeutic benefits on the SARS-CoV-2 side, this could also be an instance of um, learning about lipids and viruses together, right? Like how viruses use, because I think when we first talk about the double membrane vesicles, like way back in like talking about like these pore structures that are inside of them, one of the something I remember from that was that in HCV, that's the model system in which people first looked at these double membrane things, right? Another positive strand RNA virus that needs to enclose itself inside of these particular vesicles. Well, I mean, because it's using the same structural material in the cell to build those things, maybe there's commonalities between the way it needs to interact with lipid metabolism. So yeah, this also may be just a generalizable, interesting thing to learn about in terms of like how does lipid metabolism interact with specific viruses that need to enclose themselves in membranes for replication.
Yeah, and the lipid metabolism, if I recall, just from my own experience, tends to be a lot more harder than, say, I'm trying to understand the protein, simply because like the tools we had are getting a lot better now,
Mm,
I think.
mm.
But it's something that's so hard and expensive to
Yeah.
try and figure out. So
I'll say I have
yeah,
definitely I have
very
no
interesting.
idea on the relative difficulties of all the different metabolomics things, right? You can choose any
Mm.
macromolecule in the cell, and there's probably a suite of tools used to look at every single variant of that molecule. Yeah, I actually have no idea. It's like a very biochemistry approach. Yeah, I'm just not familiar.
Yeah, but yeah, I think that brings us to the end of our SARS-CoV-2
Hmm.
papers, and we can talk now about next paper, which is intestinal inflammation alters antigen-specific immune response to a skin commensal.
Yeah, this is a pretty dense paper in terms of the immunology, but the take-home point is quite simple. That there are it's known that there's these tissue-specific immune cells, effector immune cells, and like those are important in places where you don't want widespread inflammation every time you encounter something foreign. So that phenomenon is called tolerance. I think we've spoken about it a little bit before on the show. And so in this paper, they are looking at a specific phenomena where if you get like a immune reaction, an immune reaction on the skin, that might be somehow influencing the tolerance in the gut for immune things. Or if you like, let's say you also have like some inflammation in the gut, it could alter the way that then your skin responds to some sort of the same sort of immune the same sort of antigen.
Yeah, this is interesting because I remember like so what we refer to as often as like mucosal immunology, the immune system at surfaces. So
Hmm.
we think so skin is our external surfaces, gut is our internal surfaces, and there has been this thought that they could be linked. I remember like thinking about it with terms of like mucosal vaccinations. The idea that if you there's this idea that if you vaccinate one part, it can vaccinate all of it, the other rest of the system. But between those two compartments, it's something that this paper does a lot to kind of try to like figure out.
Yeah, they have two models, or they have two ways of interacting with the mice. So it's mouse-based, right? This is like mouse immunology as a proxy for vertebrate immunology, human immunology. We just have like a lot of control over the mouse system. So they have a way of feeding mice like DSS, which is like some irritant that causes inflammation in the gut. And so like that's a way that they can sort of cause, they can cause some non-specific inflammation in the gut and see, and then they can test the cells, right? And say like, oh, does that make the cells move around? Does that change like the specific, the antigen specificity of these immune cells? So that's one tool they have to sort of perturb the system. And then the other tool they have is like tape stripping, which is like when you just take very sticky tape on the shaved mouse back, and then you kind of use it over and over again to remove layers of cells. And then I think they put some staph epidermidis on that surface. So sort of bring the staph epi closer to the mouse skin surface. That's another way of tickling the immune system, right? Causing a little bit of like, like a local immune reaction. And the staph that they're using is like this, it's also a model in a way, because it expresses a specific peptide that is just very immunogenic. So they're able to track, the idea is that they want to track the specific immune cells that recognize that staph epi. And they can look at if those cells expand or if they decrease, but also if they're moving. Like if those cells were first, like let's say, created in the gut, they could see them move into the skin and vice versa.
Yes, that's right. They do some very clever dye technology to try and label them and see whether they. I think that the dyes like change depending if they something called Kikumi green, which upon like exposure to near UV light changes from green to red, so they can tell when something has gone to one place and then back to another.
Yeah, they have a sort of a multiplicity of tools, and the dye is also is one of them as well, to be able to see them moving back and forth. I think they also look at, there's another model system in the very same way, this like staph epi, or this Staphylococcus epidermidis that has the 2W peptide. There's also one that has ov albumin. So these are just like, this is like an interesting immunological model where I guess it's like difficult to like find a specific antigenic signal from like a whole bacteria, but with these artificial peptides, it gives them a way of tracking where those immune cells, what those immune cells recognize. And otherwise it's a lot of immunology details that I don't think I'm explaining very well, like in overview. Like they do a lot of very specific things. They also interrogate the requirement for like interleukin beta. So they're also looking at like an interferon response, an immune signaling response that governs this particular crosstalk.
I think that's it. I mean, if you want us to delve deeper into this paper and learn immunology, then just add us in the comments.
Absolutely.
But with that further ado, we'll go on to our next paper, which is Nanoscale Resolution of Microbial Fiber Degradation in Action. This is an interesting one because it's about one of the hardest things. Cellulose is one of the most abundant like forms of carbon that around there people have been trying to figure out like cost-effective ways to degrade it, and this one kind of looks into how bacteria degrade it by using one of my favorite techniques, which is cryo-EM, where they literally look at these cells in situ and see this kind of cellular machine called the cellulosome, and try and put together how it works.
Totally. Yeah, really fascinating. They use cryo-EM, so and they even point out in the methods that when they look at cryo-EM, the structure looks a little bit different than if they fixed it and didn't use cryo-EM. So this is another instance, I guess, of just the amazing potential of this particular technology. I mean, it's not a very accessible technology because I think they use machines that are like the size of a room, essentially.
Yeah.
And yeah, they're very complicated, like sample preparation machines, but they do give us this more realistic view of what the proteins and things look like, because they instead of fixing, they just like freeze things in the water. I think we've talked about this before. The freezing method makes vitreous ice, I think is what it's called, right? It's like unstructured ice, so it's not even like ice crystals. It freezes so quick, it's just disordered ice. And from that, they get a very realistic view of what the proteins look like frozen in configurations on the cell. Yeah, and they identify that there's the cellulosome, these enzymes that degrade cellulose. They're all like far out from the cell. They further than so there's the cell wall, and then also some gram-positives have a S layer, which is another set of proteins on the outside, and then this cellulosomes are even further out, and they're attached to like huge scaffolds of proteins. They're very dense.
So they're actually outside the cell. Yeah, they.
Mm-hmm.
And so they build these models based on that to show like what the structure, so they can be high density or low density depending on the situation. So I think you can see them building that 3D model of so you got in green, I think there's a crystalline cellulose, and in orange you've got the cellulosomes, which are breaking down that cellulose. So
yeah, and that's basically what's being shown here. It's like the first, again, this is like a very microscopy driven, being able to see specifically like see specifically the area in which this digestion occurs. They add in cellulose, and they can also see the cellulose strands kind of get trapped in this outer area. And they show like a relationship. So because they can see this layer now, they show a relationship where the number of cells in a population of bacteria that have more of the cellulosome on the outside changes depending on how much cellulose there is to degrade, and like when you degrade the cellulose, it releases free sugars. So it's a way of getting carbon source where you wouldn't have. I mean, that makes a lot of sense. This is for bacterial survival and their digestion. So it's interesting to see like the trade-off in population. They're able to show. Actually, if we can go to figure 3, there's just like a really good diagram there that shows it in 3B, right? That yeah, as over time, you get a shift in population to things that have more high cellulosome density that frees up sugars, but then that allows the population to switch back. So there's probably some energetic cost with maintaining this sort of cellulosome outside layer, and that's a common good within the population of these bacteria. Yeah, I think they also look at something about like where they think. Actually, I think it's through EM. They sort of show like where on this fiber the cellulosome is working on. Like it like chews up the ends or something like that.
yeah.
But yeah, just like a very interesting observation. It's also a structure that I'd never heard of before. So always I love learning about the diversity of different structures bacteria have to do specific tasks. And here's another one that's now on my radar: the cellulosome.
Yeah, and I think there's another paper. So the next paper also goes into a structure that I didn't really think too much about. Called, well, the title of the paper is "The Giant Staphylococcal Protein (Embp) Facilitates Colonization of Surfaces Through Velcro-like Attachment to Fibrillated Fibronectin." So fibronectin is this ECM protein. It lives on the outer membranes of lots of like human cells. It kind of binds things together. It's a very structural protein. It can come in 2 forms: globular or in fibrous form. And it doesn't bind in the globular form. And they're wondering and it binds in the fiber form. And they kind of, based on that one interaction, they use this paper to tear out exactly what's going on there. So they found out where the binding sites are, because the idea is that when it's outside in like fibrous form, you've got all these binding sites are available, but when it's in globular form, it kind of clenches, and all the binding sites are like facing internally. So this
Uh-huh.
protein doesn't bind to it. So you've got your Embp, which is trying to bind to all your lovely fibronectin. Can't do it in globular form. Can do it in. But then they went further. So they use this technique called atomic force microscopy, which is it's actually completely different from how you'd expect microscopy to go, because what works on like a lever system where there's like
Yeah.
it's like a record like
I,
needle, right?
It is. I think there specifically, it's like the feedback in motion makes some electricity happen that lets you see like the distance between that tip, yeah, like that record tip, the record needle tip, and the surface of something. I think I don't know if we've talked about AFM data necessarily, but we've talked about like they've used that AFM apparatus to like poke cells. I think we saw like
Yeah.
the microinjection thing sort of use like an AFM apparatus to get in there.
Yeah, they're very versatile, and that's even in more evidence here, because what they use it for a number of different things. So I think here they actually take some of this EMBP protein and they figure out that it binds. And another thing they figured out was, oh well, if you have more of it, it binds better. So they did these experiments where they dangled off like strings of your EMB protein and added the fibronectin next to that, and they've got these little force diagrams that show like when it binds. So effectively you've got so you see like this big like kind of peak, and then you can see how it kind of binds up over time. And what they kind of figured out based on the data they get from that is that it binds sort of like a zipper, where you get these long strands of EMBP which hang off the surface of the staph, and they lock in bit by bit, and they detect that kind of motion and extrapolate how this protein actually binds in real time.
Yeah, it gives the data is essentially trying to the data is essentially trying to explain why is this giant protein so giant and have so many repeats. And the idea is that, oh, actually it's because each single domain like does a little bit of binding, and if you get more of them, then it kind of zips up together like a Velcro. Going back to the title.
Yeah, like Velcro. One Velcro hook, not good. But once you have a lot of them, the bigger your straps of Velcro and whatever they're attached to, the harder it is to pull apart. And that's the idea here, where the Staphylococcus like kind of bacteria that are covered in this Velcro that likes to bind to the surface of your cells. And the thing that they're very interested in here is also like quite a lot of implants, in order to make sure they are compatible with humans, are covered with fibronectin. So it's very important to understand this when you've got those implants, because then that can make them more susceptible to staph infection. So
what I liked inside of the discussion that they highlighted in this paper was that they were saying that sometimes it's thought that like we should make implants the surface like resistant 100% to 100% antibacterial, but having like a more specific understanding of what it's supposed to be resisting might be helpful to designing those surfaces. And so here they go into more detail, like the specific conformation of the fibronectin that it binds to, or the fibrinogen.
It'd be fine. Well, let me double the fibronectin.
Fibronectin.
Fibrinogen is the pre-version of where that's like kind of
right.
The fibrinogen produces fibrin. That's why it's called fibrinogen.
Right. Right. Yeah. So this specific fibronectin, like the fibrillated form, right? The uncovered form. That's what's driving the specific binding, and so it just gives us specificity to understand what we're designing for, right? Because it's one thing to like sort of hand wave away and say that it binds to this protein, but the conformations really matter. I do feel like these might be like cellular biologists who are trying to like shed some light in a microbiological system, because they sort of make this generalized statement that maybe microbiologists don't think necessarily about all these specific receptor conformations, but inside of mammalian systems, like it's a very common like concept to be thinking
about. They come into our field to put us on blast. How dare they?
Yeah, I did think that was kind of hilarious.
Go to cell biology and leave a doggy bag of poo in front of their doorstep now.
But yeah, nevertheless, I it's useful information. I'm glad that they were able to contribute.
This is a great paper. I mean, yeah, joke's on them. They've helped our field to advance. So who's the real winner? Yeah. Next paper is Peptidoglycan recycling promotes outer membrane integrity and carbapenem tolerance in Acinetobacter baumannii.
Yes. This is like an emerging pathogen at Acinetobacter baumannii, gram-negative. And the biggest issue is that it comes up as being antibiotic resistant, specifically this carbapenem tolerance. They're going to use meropenem as they think as the representative from this group. And yeah, I mean, understanding why some bacteria are resistant to antibiotics is like a huge issue. And understanding more about that can give us, again, tools that we can use to hopefully make better antibiotics. Or, you know, there's also a really interesting realm of therapeutics where you give like an antibiotic plus something that sort of like potentiates the response. And so, yeah, so hopefully the information that's coming from here is going to help us with that. They have this observation that they're working off of where if you treat the Acinetobacter with antibiotics, some of them die, but then when you remove it, some of them can grow again, right? So they look at specifically this formation of a protoplast. So they notice that when they treat them with this antibiotic, they got kind of spherical instead of rod-shaped. And one of the things that holds the size of bacteria in check is the peptidoglycan. So a reason to become spherical would be like somehow the peptidoglycan is losing its structural integrity briefly, and then it goes back, then it comes back. And so they want to investigate that a little bit further. They do like I think they do a transposon screen. Well, they do like an RNA-seq thing, but then they also do a transposon screen just to get at what are all the genes that are changing when treated with this. What are all the genes that are changing in Acinetobacter when they treat with this particular antibiotic? And they pull out a bunch of different examples.
Yeah. Like a whole load of like different genes that, I mean, a lot of outer membrane proteins that do different things. So things that are related to stuff that comes into the cell. So when you've got things that will help you survive if you're in an antibiotic. So you want to make sure the chemical doesn't come in. You want to make sure the chemical that's in there will come out. So you've got lots of membrane pumps that are associated with resistance. I mean, this is interesting because these aren't bacteria that have resistance genes per se. These are just normal bacteria, and we're investigating this point where they're tolerating but not fully resistant, and trying to understand how it can survive to the point where. So this is like an intermediary phase between being susceptible and being resistant, and so they're looking at some of those genes to see how can we maybe stop this, make our antibiotics more effective by hitting these more tolerant like phenotypes of the bacteria.
And so one of the investigations that like in driving down like what is the specific genes that they find do is they pull out mass spec. I guess because they identify something that interacts with peptidoglycan. So they pull out their mass spec and they're like, okay, well, what could this be doing like to change the peptidoglycan? And so peptidoglycan is this like really big molecule that encases the whole cell. It's a big carbohydrate that has amino acid cross bridges to sort of strengthen that whole thing. And when they do their mass spec on it, they can see like there's a different proportion, I guess, in the mutant of these specific amino acid cross bridges. So that's their explanation for why the peptidoglycan goes away, because there's some interaction there's some interaction that depletes like the pool of some of these cross bridges that are being made that prevents maybe the peptidoglycan from being regenerated properly. and that's adaptive. that's like being able to interfere with that yeah, being able to interfere particularly with that process is somehow, well, it lets the bacteria survive briefly the excursion inside of the carbapenem. But then once that's removed, the system can turn back on and save it.
Yeah, I think that sums it up. I mean, they do highlight some proteins that are really essential
Other
for
things,
that.
yeah. I think if you go to figure 6,
Yeah.
it's like quite a complicated diagram, but like it's not just that peptidoglycan cycling they find. They also, like Omp-A is one that they point out. Like so everything in red here is the stuff that they find in their genetic screen and their translation screen. Yeah, and so like there's a bunch of different reasons that they that this could be the reason. There's a bunch of different factors that could be responsible for this antibiotic resistance. So it's like as with so many things, right? Like there's many things cooperating together in a bacteria in order for it to exhibit this like complex behavior or the resistance against antibiotics.
Yes. Speaking of antibiotics, here's a paper that discusses a new antibiotics type. So it's called Comprehensive Analysis of PNA-based Antisense Antibiotics, targeting various essential genes in uropathogenic Escherichia coli. So this is interesting for me because I was just thinking, because what they create here is antisense peptide nucleic acids. So they're like RNA, but instead of like having that sugar backbone, they've got a peptide backbone. And they've in this paper they test a raft of different versions of RNAs targeted to different genes, and to figure out which ones are performing better or worse. And they're looking at different ways to deliver them into cells. So what we have is, so let's pull up the. Again, this is quite interesting because this is essentially using the same kind of methods, trying to hack the genome of these bacteria to try and find vulnerable areas. So, and it's interesting because it's a kind of a different way to approach antibiotics than before, because I think the key thing you want to make sure is that we want it to be specific. So, because these can bind to any RNAs, but so it could even bind to human RNAs if you're not careful. So they have to be very careful about how they choose their sequencing, making sure there isn't any off targets. So
yeah, it's really weird. I've never heard of this before, PNA, but it has the nucleotide bases, so it can make base pairs. But the backbone of it, instead of being like the deoxyribose sugars, is like a peptide backbone of some sort.
Yeah.
Mm-hmm. Yeah, and the way they explained it in the paper is that lets them do tighter binding to DNA, or yeah, it lets them do tighter binding to something with a sugar backbone because the charges are different. Yeah.
Yeah, I mean that's really interesting, and because the thing they do really want to block the expression of the genes, so I think having that tighter binding is really helpful for them. So it's not like for say the mRNA vaccines where you want the mRNA vaccines not to bind too well. You just want them to be good enough to get to a ribosome to translate into protein. This one they don't want to care about protein. They just have this very kind of short length of RNA or PNA that's there to bind to the genome and to stay there to stop anything else attaching to it. So that gene is completely knocked out. So they look at the ACPP gene. They knock out a couple different ones, and they look at different kind of things to attach it to that will deliver it into the cell. And yeah, they do a lot of interesting.
So they start with ACPP, but then they go in and they try a bunch of other ones as well.
Yeah, they expand out. So I think they try to figure out which ones are best at killing. So I think they've got the ACPP here, but it's tied to different proteins or like kind of protein sequences.
No, yeah, no, they do ACPP, and then so that's like all the way to figure 4 is like just them looking at this one particular PNA that's specific for one particular gene.
Yeah.
And then later in figure 5 they expand it out to like a whole bunch of different genes. And so like I think the idea is that like if you suppress like some important gene in the cell and you prevent it from being translated, then that would just like kill the cell. that's the logic thought process that goes into selecting these genes to be suppressed. But I think what they end up finding is that's not always true because they have like a test system where they just have like a plasmid making this gene, and so they can see how well does one of these PNAs suppress that gene expression, and the efficacy at which it suppresses the gene expression is not the same as the efficacy to actually like prevent the cells from growing.
Yeah.
It doesn't correlate. Like you have to be able to bind to shut down that gene, but the ability to shut down that gene doesn't correlate to how well it kills the cells.
Yeah, I think that's an important point, because I think some genes you think you can shut down easily, like FTSE, that's an important one for cell division. And you think if you shut that down, cells won't be able to divide. But there's some redundancy in bacterial systems, or sometimes there's lots of copies. That's why you need to go out and check these things. And I think that's what this paper is doing. They're trying to kind of have that scattergun approach of trying as many things as possible to see what works.
I was gonna say, it's very strange. I guess it's like a generalized stress. I think the way that they end up describing this is that maybe it's triggering like a stress response, right? Like a backing up of the translation process, and that backed up translation process is what maybe ends up making the cells not as fit.
Right. Yeah. Yeah. So, yeah, I think that kind of sums up what I was thinking of for that paper.
Yeah, I'm not sure if we want to have more discussion. So Jackie said like not understanding what we're saying. I'm not sure if it's about PNA or it's about like the parallel chat about about SARS-CoV-2. But yeah, like it's an interest. It's a strange. Like I think that this is like a really interesting new antibiotic type, or like yeah, new way of potentially disrupting bacteria. They do one more. The other one thing that I want to highlight about this. The other one thing that I want to highlight about this particular type of molecule is that it says that the shorter the fragment is, the better it binds.
Yeah.
Which is not totally like that. You wouldn't predict that necessarily from the outset. So there's definitely like a different binding dynamics thing that's going on with these PNA molecules.
Yeah. That's right. Okay, right. So I've got my next paper. I'm queuing up a PowerPoint. So I think I just want to thank our commenters for bearing with us, because some of the stuff is quite
yeah.
Even for us, it's quite difficult. So
yeah, we're gonna say that like it's not like we do want to do some Q&A and stuff, like, but like if and we can try to work it out with you if you have like a specific question about one of the papers that we're covering. So yeah, but otherwise I do agree, like it's a little bit we're just trying to like move through and give people like a little bit of a summary of these papers. If not everything sticks, then unfortunately that's how it is. But yeah, leave us some detailed comments and we will try to get back to you. So this paper that Faz is bringing up, I'll do the intro for it. It's called Magnetically Steerable Bacterial Microrobots, Moving in 3D Biological Matrices for Stimuli Responsive Cargo Delivery. So this is like definitely like a technology, an engineering type outlook, and yeah, Faz brought up the first figure, which shows them what they made. The idea is that they want to be able to target drugs to a specific site, and the way that they're going to bring the drugs there is in nanoliposomes, and the way that they're going to move the nanoliposomes is by attaching them to essentially what is a motor, a bacteria that has a flagella that's able to swim. And how they're going to steer those bacteria is by attaching little magnetic nanoparticles of the bacteria so that they'll follow a magnetic gradient.
Yeah, so yeah, essentially they take a bacteria and they stick stuff on top of it. So they're making it magnetic by covering it in magnets, and they're adding these nanoliposomes, which can be activated by heating up slightly, or by making things slightly more acidic. And I feel like they are stretching the term microrobotics of this, because it's essentially the equivalent of me adding a broomstick to a cat and then saying I've got a Roomba. I think that's kind of the
Yeah, for sure. Yeah, like I've seen this sometimes on social media, like a Roomba that someone like attaches like a knife to make a battle bot.
Yeah, but essentially they've got a living thing that does its own thing usually, which is quite good because bacteria can do complicated things, as we've talked
Hmm.
about previously. And so attaching like various, this could be interesting for thinking about treatments or other things like that. Um, and this is a really interesting paper because they got like a lot of videos and stuff.
Like I feel
And
like we
they
might
attach
have.
these nano.
I was gonna say we might have seen nanoliposomes being used to deliver drugs at some point. I know that people attach antibodies to nanoliposomes,
Yeah,
right? And you can then target via antibody this object. But if you just do an antibody, that's just relying on diffusion. It doesn't. It can't like have its own self-propelled way of getting places. So, yeah, the idea is that with some propulsion, you can get into spaces that you wouldn't otherwise be able to get into.
yeah. And in this case, they're carrying doxys doxycycline, I think, or doxorubicin. I can't remember exactly, but it's a
Yep.
chemical that we want to deliver to a cell, and so they've got these little nanoliposomes that when they have this thing in the membrane, so this fluorophore, so it absorbs light and converts it into heat. So this one converts infrared. So if you stick an infrared laser on it, it'll start to heat up a lot more than say a normal fat. So therefore you can pop these liposomes by pointing a laser at them, which a low-powered laser I have to point out because if you point a high enough power laser at anything, it's not going to be happy. But I think
Right.
they're getting this at the sweet spot where it'll pop where they want it to.
Yeah, I think
Um,
I think there's something inside of these nanoliposomes that makes them responsive to the laser light. Yeah,
yeah,
a
so
specific
that's
dye. Yeah.
IgG, so that's green in these top left diagrams,
Hmm.
and that is supposed to absorb the light, and that's supposed to make the liposomes like open up when the light shines upon them.
Hmm. Hmm. Yeah, so quite a complicated, quite a complicated process. But what they try to show is that it really does get. So they use organoids as their test material. And so they show that, oh yeah, like actually when you bring these bacteria and have them swimming around, they can direct them down one of two channels. So it's like not about, it's not diffusion based. The idea is that it's not, they don't just go everywhere. They go where the magnetic field is pointing them towards. Oh, there's a movie. This is
Yeah,
interesting.
there's a
Yeah.
movie, so we've got a couple of movies. That's why I've put this in PowerPoint form. So they've got so firstly the thing they want to make sure is that when they add these biohybrids, these like kind of liposomes onto these, how whether it they have to make sure the bacteria are okay, to
Hmm.
make sure that, because obviously if you bacteria, they're living organisms, you stick to stuff to them, they might not like it, they might end up going, and they do find that when you attach like magnets to them, they do swim a bit slower,
Hmm.
uh because again that happened to me if you stuck a bunch of magnets on me while I'm swimming. um but that's but and let's see, and let's see if I can pull on to the next video. uh Hold up. And also like so they also see like what happens when they add a magnet. So now you've got these magnetized bacteria. What
Hmm.
happens if you add a magnet to them? So we've got these bacterial hybrids that are now swimming across a magnetic field. So now you've got these bacteria that are there's some actual force pulling upon them, and you can see they're going a lot
Mm.
faster.
Yeah, and they're also going in like a directed line.
Yeah, they're going across the magnetic field lines.
Hmm.
So again, I'm not sure exactly how the magnets are polarized, but I think that motion makes it easier across field lines than through other things. And you can they do some fancy movement with them. Uh, so one thing they do is like now that you can use these magnets to steer the bacteria around, to move them around.
Yeah,
Um,
it's wild.
so we've got some video of them moving bacteria in a square shape using these magnetic fields.
Hmm.
Uh,
And I think that they're thinking about a cancer model here, where they say like, right, imagine like there's a tumor somewhere, and you want to infiltrate that tumor with a bunch of these bacteria that are carrying a drug specifically to destroy those cells. Like this is, you could somehow, because people do imaging to know like, oh, here's the tumor in a body. The idea is that if they have this amazing control over a magnetic bacteria, they could use those magnetic fields to direct the bacteria to swim into your tumor.
Yeah. So, and in fact, that's
Certainly,
what they do.
going in a square is a sort of far away from being able to maneuver inside of a body.
Well,
Um,
yeah, I think that's more like a test. So that's your
yeah.
bragging thing. You do that to test the technology. So you get that
Hmm.
sometimes where you get like atomic, where you get like people etching their names really tiny or things like that. I think
Yep.
disapprove the technology,
Yep.
and so now if they can do that kind of movement, then being able to move it towards spheroids should be a piece of cake. So they've got this setup where they got a couple of like, well, the bacteria in one vessel, some tumor spheroids on another, and then trying to steer them towards a specific tumor spheroid.
Hmm. Yeah, I think to me it's this final figure four, that like brings home like why this might be important, is because they're able to they're able to get these swimming bacteria through like a tighter matrix. So they make like different stiffnesses of collagen gels, and they can get them to go further in those gels than they would have otherwise been able to go just by diffusing by themselves, or even just by swimming. The magnetic force, the directed magnetic force, is actually helping them get through like a denser gel.
Yeah, I've got some. So I'm going to skip past
Oh,
these
sorry,
videos
you
to
got a lot
talk
of
about.
images.
Yeah, but so I mean, they got do a lot of things. They also like try and mimic veins in their little
Mm.
stories to try and simulate. Okay, well, how can these move under flow conditions? So let's say you got a blood vessel where you've got lots of liquid flowing through in a direction. How can these hybrids flow through towards the
Hmm.
tumor? So now you got an image of these tiny bacteria being steered directly into a tumor here.
Yeah, so here I think flow is going perpendicular to the magnetic field. So the idea is that
Yeah,
you can pull the organisms out from flow and pull them into, like, yeah, wherever that tumor happens to be.
yeah. And here's a diagram of the collagen
Oh yeah,
gels
that.
where they've
Mhm.
got these blocks of like collagen that get thicker as you get. So I guess it's hard to visualize if you haven't worked in a lab, I guess. But uh, I'm. But yeah, I think it's just like
No,
these thick
it's like a
mounds of
triple
jelly.
triple layer Jello.
Yeah,
like like those like Jello desserts that have different layers, except in this one the Jello is getting more and more firm. And yeah, and they just see how far can these guys diffuse through, and the presence of the magnetic field gets them into the densest of those gels.
yeah. So here you've got like I think they have varying stiffnesses. So I think the way to think of it is almost like as you get stiffer, the holes get smaller, and it gets harder for the bacteria to get through. Um,
Uh-huh.
and the magnet kind of produces that extra force that pulls them through to the place where you want them to be. So it gives them a little bit of help. Um,
Mm-hmm. Mm-hmm.
yeah. Let's see. So here we're just basically watching bacteria. Well, that was trapped, but now he's escaped. Yeah, you see them. They're stuck in this particular area, and then how are they going to get through? They they somehow get through. I guess they wriggle their way out, but
Is the alignment correct on this one? It's like saying the alignment is in the other
well,
direction.
thing about magnet fields is like that's how we say the alignment goes, but the thing is like the
Oh, it doesn't. Yeah, depends on which side it's actually being pulled towards. Yeah.
yeah. So what that mostly talks about is like people like kind of iron filings go along the field lines, but the actual
Hmm.
direction it can go either way. It just depends on what charge that not charge, but what kind of
Right.
polarity
Right.
that bacteria
Right.
has. So if the bacteria has the right polarity, it'll go in one direction, or if it's got the opposite polarity, it'll go in the other direction.
Hmm.
And I think that's basically and yeah, that's the big thing about this. I think the idea of like getting these bacterial cells to swarm around these tumors and deliver a kind of a little package of of drugs that will kill the
Yeah.
tumor.
I think you also have to remember here, like in terms of like how far is this technology along to being an actual therapeutic, you have to think about like I'm not really sure about having like like bacteria in your bloodstream, like the LPS has to be changed so it doesn't like cause inflammation and all sorts of things. Like also, will these objects snagged by your immune system, being like, oh, we don't want these random swimming things around. Let's just like take them out of circulation.
Yeah.
But yeah, but we also covered. It also reminded me that we covered a paper a while back saying that like there are some flagellins in the flagella that are non-immunogenic if they're made from the commensal bacterias. Like so, there may be spaces where there's some like we can choose proteins that have evolved with commensals in us, and then be able to make these bacteria more like a commensal platform. Yeah.
Yeah, I mean there are a couple of things. This is a great proof of concept. I think there's lots of interesting applications out there, but for actually going into like a hospital and having a magnet put on you and hoping that all the bacteria from this come to it, it's going to be very difficult because there are a couple of technologies here that they rely on something called the inverse square law. So as you get further away from a magnetic field, it's the kind of strength of it decreases by a square rather than linearly. So as you get further away from it, it becomes way less potent. So something that will work in a petri dish might not work as well
Yeah, that too.
scaled up to the size of a person. So this is a particular thing for things like light. So humans, as you can see, we're not entirely transparent. We're quite opaque. So whenever you have a technology that's based on light shining through us, it isn't as effective as it would be through mice. Because even though mice are also opaque, they're smaller, so you can use more powerful light to get through. Whereas since we're much larger, it's a lot harder to do that. I feel like something similar happens with magnets, where you have to use a very strong magnet, and I think that could be possible here. But yeah, it's hard to imagine at this
They built a
point.
very interesting, complicated machine. Like, yeah, it's very. I have no idea what the actual applicability is inside of a human.
I think there's a lot of things for if you're thinking about, like, say, I've got a cell, let's do some surgery at a organelle level at the top. Then maybe having something like this could be very useful as tweezers, or I think, and even like steering bacteria and using that as, say, if you've got like a a bioreactor, you've got lots of different species of bacteria that have to do differently, and then you can use these to kind of manipulate some.
Oh
So
yeah, that'd be interesting. Like spatial regions within a certain a certain space that like do different
yeah,
chemical reactions.
because in a
Yeah.
bioreactor, you're creating almost a bacterial city with different bits of the community. So
Uh-huh.
so using
Uh-huh.
robots to control that could be an interesting way to. I mean, there's lots of other. This is one of those
Yeah.
technology where there's so many other applications that you can
Yeah.
think about. But
Yeah.
cancer is kind of the easiest one to demonstrate like applicability. If you can
Yeah.
get more money by saying, "Hey, this
I was
can
gonna
cure
say
cancer," than saying,
it also sounds real sexy.
yeah, rather than saying that this is a technology that can change. We can't even think of this. It opens so many doors. We can't even think about what doors it opens
Right.
at this point.
Right.
It's
Right.
much easier to like kind of pare it down and say, hey, this can cure cancer.
All right. Last paper then.
Yes, last
Not this
paper
one.
is measles and Nipah.
Measles. Oh,
Sorry, you can say it this time.
okay. Measles and Nipah virus assembly specific lipid binding drives matrix polymerization. So here it's actually a big structural biology paper looking at this M protein, the matrix protein embedded in the membrane of these envelope viruses, and the way in which I guess it contributes to budding, and specifically it has to bind a lipid in order to initiate this process.
Yeah, so big part of the creation of viruses is the point where it's just about to exit the host, and at that point they're actually creating the structure of the virus. That's called the budding. So you've usually got like a lipid membrane where it buds off, where it kind of like forms a shape. So it needs to be able to package everything within it. And so in order to do this, there are various proteins there that need to make sure it's the right shape, because if it's the wrong shape, then it may not fit in the right areas. So I think because I think the virus shape is very important to some viruses. And this is and so we've got your membrane, which is made out of lipids, which are fats, and this one looks at how the M proteins bind to some fats. So specifically these PI45P2. The names aren't necessarily that important. The important thing is to understand there are specific things in the membrane that that you can bind. And I think the image that I think best kind of illustrates this is in Figure 3C, where you've got your viral protein that's got this charge, and then it binds. When it binds, it changes shape and it bends outwards, and then it bends the membrane around it. And so this paper is essentially trying to study what's causing that, because it could be an electrostatic interaction. Is it because of the charges on the protein, or is it because it's doing some specific binding? And I think what they find is it is specifically binding a specific chemical component on the membrane, and using that as anchor points to kind of bend the membrane around it and make it more spherical.
Yeah. And they find the structure. They do mutations to like mutate that binding pocket so it can't bind the lipid anymore. And then if it can't bind the lipid anymore, then it doesn't do the membrane budding. It doesn't do the budding or the curvature as much. And they even have like, and they determine like a structure of this. I think it's a dimer. Yeah, they do a structure of the dimer where it has the lipid inside of it, or it doesn't have the lipid inside of it, and they can see that in those two formats it has like a different shape.
Yeah, so when we mean dimerize, it pairs up with other molecules that look like it. So it kind of forms a structure. So this isn't just one protein that's creating one little dimple. It's a number of proteins that are creating an arc that turns into a circle. So
yeah, they tested out on these giant. We've seen these things before. The giant unilaminar vesicles. In Figure 7, they have like a nice image. That's like a nice functional way of seeing whether or not this stuff is working. They can like change the composition of those vesicles, and if it doesn't have that particular phospholipid, then the bending doesn't happen.
Yeah. So So this is quite neat because it's something that you can target. So once you know that a key part of a virus's metabolism that's separate from the host, you can target that. And if you can stop a virus from budding, then you literally stop it from being born, which is something that we want to do. As we don't want viruses to spread. So anything more you learn about viruses gives us a new way to combat them.
Yeah, like I think they point out that this protein is, I guess, in common with these paramyxoviruses. So yeah, measles is one and Nipah is one. So yeah, so something generalizable here about viral biology.
Yeah, I think that brings us to the end of our papers. But yeah.
Did you want to say something about the prevalence? I feel like the chat that was had in the thing that we had in the chat was like just saying like SARS-CoV-2 is everywhere.
Yeah, it is everywhere. It's it's one of those things where now with it is that there isn't much. We now know what the issues are with SARS-CoV-2. We know how to protect ourselves with, and it's been. I mean, you recently got SARS-CoV-2, Danny, right?
Yes, yes. Yeah, and I was thinking based on the timing of it, it was BA2. But again, like just to go back to the point that I covered in the chat, like I think that the reason that that's just like from looking at the rates in my area and looking at the genotypes that I got associated. But of course, like I don't have like a confirmatory genotype to say that that's what I got inside my body. So like I have no idea actually in that sense. But I have a good sense that it might be that I probably got BA2. But I know, and I also think I know why, because like I went to a conference and I had a meal. I had a bunch of meals unmasked. So like, you know, there's like a clear sort of correlation there. Yeah, I don't know. This is tough because, you know, at least in the news, we see a lot about like living with SARS-CoV-2 and what does that mean. And like it's tough because it's everywhere, so people have to be careful in all these different areas. Yeah, that's I don't know. That's what I have to say around that topic.
I mean, yeah, it really sucks right now because it has people want SARS-CoV-2 to be over and done with. But we keep kind of releasing we keep releasing the accelerator just before it's
I don't want to say we say it all the time.
Run SARS-CoV-2 and then we release the accelerator, anticipating how
Yeah, totally. Like we've talked about a lot, like the Swiss cheese model, that everything, especially in biology, not everything is like, there's no like big absolutes. You just put up like sort of statistical barriers. There's still like a small chance something could get through. And like the vaccine is like one statistical barrier, masks were a statistical barrier, right? Like different behaviors are a statistical barrier to try to limit the spread. And yeah, just as we see like the decrease in things, then we remove some of those barriers, and then yeah, we're in like a different steady state, one where the virus is a little bit common in not a good way, I think. Yeah.
Yeah, I think the main issue now is I think in terms of treating the virus, we're as good as we're going to get. But now the
Yes. Yes.
concern is about the post-COVID symptoms as well.
Yeah.
Uh,
Yeah, like
and
dangerous acute disease, hospitals are quite good at it. Of course, like it's still possible to overwhelm a hospital system, but I
mm,
think that people are watching for that and trying to sound the alarm if that happens. Mhm.
but yeah, I mean the vaccines are out. I mean, I've been vaccinated.
Yep.
Lots of people have been vaccinated. So I think the concern about dying immediately from SARS-CoV-2 is a lot lesser than when we first started
Oh,
this
oh, absolutely,
podcast.
absolutely. I feel like, and there's also the Paxlovid, at least in the States, like it is kind of common around here. So people also have like a pharmaceutical intervention they can use. Yeah, there's lots of tools to do away with the most severe, acute severe symptoms. But of course, like those who are immunocompromised, those who are like at risk of like a bad outcome, that yeah, that's the that those people maybe are a little bit of a disadvantage these days in our society.
Yeah. And also the disclaimer, we're not public health professionals.
Yeah, that
We're
too.
people off of YouTube. And I'd be really bad if I didn't mention that getting your information off of YouTube there's a bit of buyer beware there. I
Yeah,
mean,
yeah. There's lots of good. There are some good sources on the internet that you could look, like sort of authoritative public health folks, um, and and yeah, everyone has
yeah,
their own specific risk profiles. Well,
it's
we can't cover it all.
yeah, it is actually very hard to talk about this because I'm trying to think about so how I get my information about what to do about COVID tends to be because I'm based in the UK, looking at NHS,
Hmm.
maybe keeping an eye on the local public health kind of. But the thing is, I live in a place where we've got like a very uh, we've got alert public health system, so
Hmm.
we do get a lot of interactions with that, and even like going to hospitals, they literally give you a mask as you go in
Hmm.
to a hospital. I mean,
Hmm.
it's so. I mean, I don't know how it's like in the States, but I I mean, the thing is like
I don't really know
also you
either. I
can
feel like it's super diverse here. There's like,
yeah
depending on where you live, like depending on who you like associate with, people have different like yeah,
yeah
different norms are being followed. Yeah.
and yeah, I feel like really bad because I already have a PhD in microbiology, so my way of going through the wading through the information ecosystem is very different. And there are lots of unconscious things I do that I don't know. So I do worry that any advice I give people is going to be go over people's heads because it's a lot of the things that I look for, like who's talking about this news story, is like that kind of it's tools that everyone can use, but the way I use them are kind of different.
Yeah.
So
Yeah. But that's all I want to say about the subject. Do you have anything else you want to add, Fuzz?
no, I'm just want to say thank you so much for listening. I understand that sometimes we do go too much into the weeds sometimes with the techno jargon, and
Yeah.
I'm gonna try to be a bit better with that in future. But
Yeah.
thank you very much for
Yeah.
listening and
But
being part of this.
also as I said, or I tried to express in the chat too, is like if you do have a specific question and something is a little bit unclear, like yeah, you can ask synchronously, and we'll try to address it on the spot.
Yeah, that's why we're
But of
live.
course, like
So yeah,
yeah, that's why we're live, is specifically for that purpose. But of course, asynchronous also works if it kind of strikes you a bit later. Yeah, Mastodon or Twitter, inside of the YouTube chat, like we do see all those things. So yeah, and we hear you, Andrea, about like being interested about the immunology paper and immunology in general. It's definitely like a huge like. I think we both studied the host pathogen interactions. That was like the framing of our PhD
yeah,
work. So like it's a huge part of understanding infectious disease microbiology to not just look at the pathogenic factors on the bacterial side, but how they specifically interface with immune system, and not just the immune system in the specific tissue that you're looking at in the context of that last paper we studied, but like even more, right? Like the immune system globally in the
I mean,
organism.
immune tolerance is such a massive topic, and it's so important as well. Because I mean, as someone who suffers from hay fever, I'm always worried about immune tolerance, because one day I want to be able to go outside properly. And there's so many interesting things, especially with, and yeah, there's a lot to cover. But yeah, it's something that we'll try to delve into a bit more to try, because again, immunology is one of the things that does also confuse me a lot, which is why I try to be as general when talking about it. Yeah.
Yeah. So join us next week for more cool microbiology news.
Yeah, and as we've said, if you have any questions or if you, because again, oh gosh, I've lost my script, but yeah, we're just people who are very enthusiastic about microbiology, but we don't know everything. So if there's any corrections or comments, please pipe up in the comments.
Yep, absolutely. Peer review is this process of discussing the papers and really reading the papers. So that we hope that you've had a good time like listening to us ramble on about microbiology today.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Bye.
Not next week, 2 weeks.
2 weeks, yeah, 2 weeks. 2 weeks. Sorry, yeah. Right, tune in 2 weeks for more microbiology content. Bye.
Bye-bye.